Hello everyone, welcome to Meat, Fish and Poultry Process Technology. So my name is Shiva Prasad. I'm an assistant professor at Department of Food Technology, Vignan's Foundation for Science, Technology and Research. So students, in today's lecture, we will be having an important discussion on one of the important and interesting topic, that is chemical and nutritional composition of meat. So in today's class, we will be having an understanding on one of the important concept, that is the eating quality of meat. So in this particular concept, which establishes the palatability of the meat, so we will be concentrating on three palatability characteristic of meat, that is color, water holding capacity as well as juiciness. So these are the three important contents that we would be covering in today's lecture series. Okay, so before anything, let's have an understanding on one of the first important eating characteristic or quality of meat, that is color. So when we talk about color of the meat, so it is an very important sensory characteristic of meat, because it will have an impact on the consumer perception of quality. So how exactly the quality of the meat is? So it is exclusively defined by the sensory aspect, particularly the color. So if the color of the meat is aesthetically appeal to the consumer, then he or she tend to buy the meat. So color is one of the important sensory characteristics which will have an impact on the eating quality of meat. Okay, so similarly, the amount of moisture that is present in case of the meat will also have an influence. So as we are already aware, so the water will in turn absorbs and emits or reflects the light, so which will be visible to the consumers or anyone who is viewing it. So the amount of water that is present will have an influence on the degree to which the light is reflected, and thereby it will have an impact or imparts the glossiness or lightness to the final product. Okay, so that is how the color characteristic in case of meat is observed. So following which, the muscle pigment. So specifically speaking, we are talking about one of the important muscle pigment, that is myoglobin. So myoglobin constitutes to about 80 to 90% of the total pigment in case of meat. So this particular pigment is a heme containing macromolecule. So this particular myoglobin is exclusively responsible for imparting colored characteristic to your meat. Okay, so besides myoglobin, we also have certain other kinds of pigment, so namely hemoglobin. So hemoglobin will constitute to about approximately 10% depending on exsanguination. So exsanguination is nothing but how much the animal has been bled. Okay, so depending on the exsanguination, so hemoglobin content in case of meat will in turn vary. Okay, so the hemoglobin is also one of the important pigment which will in turn contribute to the color characteristic of the meat. So besides these two important pigment, we also have certain other important pigment such as like cytochrome. Okay, so cytochrome is also one of the heme protein, but hemoglobin as well as cytochrome do not actually influence or have a much influence on the characteristic specifically towards the color. Instead, myoglobin is the one, because it is contributing to the highest percent, that is 80 to 90%, in turn imparting color to the meat. So taking this into consideration, so we have to have two important factors here. So that is the concentration as well as the stability. So as we are aware, so myoglobin is contributing to the color of the meat. So how much of concentration of myoglobin and how stable this particular myoglobin in case of meat is will in turn have an influence on the color of the meat. So the appearance of the meat will change depending on the concentration as well as stability of the myoglobin existing in case of meat. Okay, so following which, let's have an understanding on how exactly this color pigment, that is myoglobin structure, exists in case of meat. So as we are aware, so the myoglobin consists exclusively of two important portions. That is the globular protein portion, which is regarded as globin, and non-protein portion, which is regarded as heme ring. So these are the two important portions that are existing in case of meat, okay, specifically the myoglobin, okay. So as we can actually see in this particular figure, so this is the heme ring, okay, so this exact portion is the heme ring, okay, and this is the globin. So in the center of the heme ring we have iron, okay. So as we can actually see here, this iron has 6 different ligands, okay, so 4 of which are bound inside this particular heme ring. So the iron shares 4 ligands and it is bound inside the heme ring. So the rest of the 2 ligands are free, okay. So one is exclusively bound to globin, okay, which is the globular protein portion, and the other one is the free binding site. So depending on what kind of compound, specifically the chemical compound, is attaching to this particular free binding site will have an impact on the color of the meat. So in the further slides we will see what are the different kinds of chemical compounds can actually bind to this particular free binding site and what exactly will be the color change that is observed in case of meat. So this is about the myoglobin structure, okay. So to revise and to give you an indication, so myoglobin exclusively has 2 important portions. So the first one is globular protein portion, which is regarded as globin, and the non-protein portion, which is the heme ring. So as you can see here, this is the heme ring, in the center of which we have iron, okay. So this iron has 6 ligands, 6 binding sites, 4 of which are shared within the heme ring and 1 ligand is shared between the globin and the other ligand is free binding site. Okay, so depending on what compound is attaching to this particular free binding site, the color of the meat will in turn change. Okay, so I hope this is effectively understood by everyone. Okay, so moving on, let's have an understanding how this myoglobin content will have an influence within the species. So in the species, how exactly the concentration of the myoglobin varies and what is the color that is imparted accordingly. Okay, so in the first figure I have shown you the fish. So the color of the fish meat exclusively varies from grey white to dark, so which can be exclusively seen in here. Okay, so this is exclusively because of the myoglobin pigment existing. Okay, so similarly we also have poultry. So the poultry meat exclusively have a color that is grey white to dull red. Okay, so similarly we also have pork. So the pork meat is the greyish pink. Okay, so following which we also have lamb as well as mutton. So the color of this particular meat varies anywhere from light red to brick red. So similarly we also have veal. So veal is nothing but it is the meat of the calf. Okay, so it is having brownish pink as you can see in the picture. So similarly we also have beef, which is imparting bright cherry red color to your meat. So finally we also have horse meat, that is dark red in color. Okay, so this particular color difference is exclusively because of how much is the amount of myoglobin that is existing in case of the meat. Okay, so that is how much the species and the amount of myoglobin will also have an effect on the color characteristic of any kind of a meat product. Alright, so following which, let's have an understanding on what are the important factors that will have an impact on the color characteristic of the meat. Okay, so the first important characteristic that we would be covering is the quantity of myoglobin. So how much is the quantity of myoglobin? So because of the difference in the quantity of myoglobin, how is the characteristic color difference observed in case of meat product? So in here we will be concentrating majorly on 3 important factors, that is myoglobin difference. So how exactly the myoglobin content exists in the same species at different age levels. Okay, so that we would be covering, and also the species difference in different species like poultry, pigs, cattle. So how much is the exact variation observed in case of myoglobin. So similarly the type of the muscle. So how exactly the muscle will also have an impact on the color characteristic of meat. So that would be, we would be covering. So following which, we will also see how chemical stability of myoglobin will have an influence on the color characteristic of meat. So finally, we also have the cooked color problems and the vitamin E feeding to the cattle. So vitamin E is a antioxidant which will in turn prevent oxidation of the myoglobin. Okay, so that we will study in the further slides. Okay, so similarly we also have how packaging material will also have an influence on the color characteristic of the meat. So these are the 5 important factors which are in turn affecting the color of the meat. Okay, so let's have a discussion on the first important factor, that is the quantity of myoglobin. So how quantity of myoglobin will also have an influence. So the first thing that we would be seeing is myoglobin difference in age, okay, within the species. So in the same species, how exactly the quantity of myoglobin is varying. So herein I have established a table for beef. Okay, so here I have given the age class as well as the myoglobin content. Okay, so in case of veal is nothing but the meat of the calf. So it is having very low content of myoglobin, that is 2 milligram per gram of meat. So similarly the calf will have 4 milligram per gram of myoglobin content, and also the young beef will also have 8 milligram per gram of myoglobin content. So the old beef will have 18 milligram per gram of myoglobin content. So it is clearly evident from this table that as the age of the species is increasing, the quantity of the myoglobin is also increasing. Okay, so that is exclusively because the muscle fiber is increasing and thereby the content of the myoglobin is also increasing. So established a table which specifically describes the myoglobin content in different species. So as I have actually shown here, so the pork, the color of the pork meat is pink and the myoglobin content is 2 milligram per gram. So similarly we also have lamb meat which is light red in color and the myoglobin content is 6 milligram per gram. So likewise we also have beef which is having cherry red color, which is exclusively because of the increased myoglobin content, that is 8 milligram per gram. So it is evidently observed from this table that as the myoglobin content increases, so the color of the meat will also turn darker and darker. Okay, so that is what the interspecies difference specifically in terms of myoglobin content and therefore the color of the meat. Okay, so moving on, we have our third important type of characteristics belonging to the quantity of myoglobin, that is the type of the muscle. So what part of the meat is exclusively used for consumption will also have an impact on the quantity as well as the color characteristic of the meat. So herein also I have established a table which specifically describes what is the type of the muscle and the name of the muscle as well as the myoglobin content. Okay, so the first is the locomotive muscle. So whenever we are using locomotive muscle, specifically the extensor capri radialis, okay, so it is having somewhere around 12 milligram per gram of myoglobin content. So herein I want to show you a picture as to understand what is the color characteristic of this particular locomotive muscle and the corresponding myoglobin content value. Okay, so when it is 12 milligram per gram, the meat will be extremely darker in color. So similarly, we also have support muscles. So whenever we are consuming support muscles, specifically the longicium dorsi, okay, so the myoglobin content in this particular muscle is 6 milligram per gram. So here also I have established a specific picture so that you will evidently differentiate between the color characteristic in both of the type of the muscle meat. Okay, so in case of locomotive muscle, the color is darker, fine, in corresponding to the support muscle, which is lighter in color. Alright, so this is about the type of the muscle. So moving on, let's have an understanding on the second important characteristic which is affecting the color in case of meat. That is the chemical state of myoglobin. So how exactly the myoglobin is existing in case of meat will also have an impact on the color of the meat. Okay, so herein the color is dependent on myoglobin state. So how exactly the myoglobin is existing? So what are those type of myoglobin? Okay, so first in we have deoxymyoglobin, we have oxymyoglobin, we have metmyoglobin, we have carboxymyoglobin, we have cyanide metmyoglobin. So these are the important types of myoglobin which is existing. Okay, so besides them there is also numerous other varieties. So in this particular lecture we will be concentrating on three important types of myoglobin state, that is deoxymyoglobin, oxymyoglobin as well as metmyoglobin. So this is what we would be studying in today's lecture. Okay, so the first in we have deoxymyoglobin. So as I've already told you in the previous slide, so this particular myoglobin structure and what is the specific type of binding site or the type of chemical that is binding in this particular site will in turn have an effect on the color, appearance or the color characteristic of the meat. So specifically in case of deoxymyoglobin, we have water molecule which is attaching to the free binding site, and also the iron is existing in terms of Fe2+. So besides this we also have the globin. Alright, so this particular of meat will have purplish color. So as I clearly described in this particular figure, the color of the meat which has deoxymyoglobin pigment will appear purple in color. So this is exclusively because of the reduced form of iron, that is Fe2+. So this Fe2+ will have an impact on the color or the appearance of the meat. Okay, so if Fe2+, which is the reduced form, is present, it is highly appearing in terms of purplish color. Okay, so that is how the color of the meat changes. Okay, so this form, whenever it is existing, since it is a reduced form, it is highly unstable. So this particular type of ring, it becomes highly unstable, and that is the reason why they readily combine with oxygen. So when they combine with oxygen, they will get reduced to oxymyoglobin or metmyoglobin, in turn changing their color. Okay, so this is about the deoxymyoglobin. So to give you a revision, so the deoxymyoglobin imparts purplish color to your final product or the meat, and it has a reduced iron form, that is Fe2+, which is highly unstable, which goes on to bind to oxygen, and then whenever it is exposed to air, and then it reduces or changes its form, that is oxymyoglobin or metmyoglobin. Okay, so the second important type that we would be studying is the oxymyoglobin. So as I've already discussed, so oxymyoglobin, so the compound that is attaching to this particular free binding site is oxygen, and the iron that we have that is existing here is Fe2+. Okay, so this type of oxymyoglobin, whenever it is seen, so it will impart bright red color, which is exclusively seen in case of beef. Okay, so it is imparting bright red color because of the presence of Fe2+ and oxygen molecule. So the oxygen molecule is attaching to the free binding site, and thereby it is imparting the bright red color to your meat. Okay, so under atmospheric condition, this oxymyoglobin is stable and it cannot be easily oxidized to metmyoglobin, because metmyoglobin will impart dark brown color to your meat, okay, which is highly non-appealing. Okay, so this particular under atmospheric condition, it is stable. Okay, so then because it requires continuous supply of oxygen. So why oxygen is required? Because the enzyme which is responsible for imparting this particular bright red color requires continuous supply of oxygen. So as long as oxygen is present in the atmosphere where this particular meat product which is having oxymyoglobin pigment is present, then it will appear as bright red color. Okay, so this is about the oxymyoglobin. Okay, so following which we have next type, which is metmyoglobin. So the metmyoglobin, we have water in the free binding site and we have Fe3 plus. Okay, so which is in its Fe3 plus site. So the presence of this oxidized Fe3 plus will impart brown color to your meat product, which is highly unappealing. So you will not feel like consuming such kind of product. So we should not let this particular reaction to happen. Alright, so this is very oxidized, okay, which because it is having Fe3 plus and thereby it becomes very stable. So the color will in turn remains most of the time until and unless the oxygen is removed or added. Alright, so the meat remains brown in color indefinitely as long as it is exposed to air. So as long as it is exposed to air, this particular meat will remain dark brown in color. Okay, so that becomes highly unacceptable. Okay, so only by removing an oxygen, so as I've already told you, if we are removing oxygen and developing reduced condition, then we can convert it back to the desirable color. So if we are not doing that, the color will remain indefinite period of time whenever it is exposed to oxygen. Okay, so this is one of the important other type of color state of myoglobin. So far we have studied three different state of myoglobin, that is deoxymyoglobin, oxymyoglobin, as well as metmyoglobin. So in the next slide I have given you a summary table which establishes what is the chemical compound and how exactly it is imparting a specific color to your final product. Okay, so herein this is the myoglobin reaction. Before moving on to this, let's have an understanding on myoglobin reaction. So we have deoxymyoglobin. So this deoxymyoglobin is what is in color? It is purplish in color. Okay, so whenever it is exposed to oxygenation, that means whenever it is absorbing oxygen, it will convert back to oxymyoglobin, thereby imparting a specific color that is bright red in color. Okay, so following which, if we are again reducing or we are removing an oxygen, we can convert it back to deoxymyoglobin. Okay, so similarly, when deoxymyoglobin, which is purplish in color, if we are adding oxygen to it, we are expecting metmyoglobin. So whenever we say metmyoglobin, it is exclusively brown in color. Okay, so this is the myoglobin reaction in a nutshell. That is, the deoxymyoglobin in the presence of oxygen converts to oxymyoglobin. Whenever an oxygen is removed and a reduced environment is provided, it will convert back to deoxymyoglobin. So similarly, the deoxymyoglobin in the presence of oxygen or oxidation, it will result in metmyoglobin pigment, which in turn imparts brown color to your final product. Okay, so this is the myoglobin reaction. Okay, so let's have an understanding on the summary table here. Okay, so the kind of compound which is responsible to imparting a specific color to your meat product. Okay, so herein I have given you the compound and the specific color that is imparting and the name of the compound or the pigment that is being formed. Okay, so the compound H2O, that is water, will impart purplish color, and the compound that is myoglobin state that is responsible is deoxymyoglobin. So similarly, so the other type is carbon monoxide. Whenever such kind of chemical compound is binding to this particular free binding site, it will impart red color, and the resulting compound for the same is carboxymyoglobin. So this is the summary table which we have studied so far, which have established what are the specific compound that is binding to the free binding site, in turn resulting in the specific color of the meat, thereby formation of a specific myoglobin state. Alright, so this is about the chemical state of myoglobin. Okay, so following which we have another type of factors that is affecting the meat color. Okay, so the next type of factor is the cooked color problems. So this particular cooked color problem can be categorized into two different types. Okay, so the first one is premature browning and the second one is persistent pinkness. So these are the two different problems which are existing as far as cooking characteristic is concerned. Okay, so when we are talking about premature browning, so usually it is exclusively caused by the oxidized meat or the meat that is exposed to oxygen. That is, whenever we are keeping a meat product in an environment which has excess amount of oxygen, it is resulting in the premature browning. So as we have previously studied, so if oxygen is binding to the specific binding site, it is result in the color change. Okay, so thus it will impart a brown color to your product. Okay, so which is highly undesirable. Okay, so that is about the premature browning. So similarly, whenever we are subjecting the meat slice for cooking, so the cooking will also have an impact on the color characteristic of meat. Okay, so when we are cooking at a temperature of somewhere around 55 degrees centigrade, so the surface of the meat will appear brown in color. So in this particular picture I have evidently shown you. So in the figure 2 you can exclusively see whenever we are subjecting the meat for cooking or grilling, so the color of the meat is distinctively changed whenever 55 degrees centigrade is reached, but the inside temperature remains significantly less. Okay, so thereby the meat will appear pink in color. So this is exclusively dangerous because it is posing a food safety threat. So why exactly it is? Because the pathogenic microflora that might be present in case of the meat will not be killed. Okay, so which is one of the major problem as far as food safety is concerned. So whenever we are heating the product, specifically the meat, or grilling it, when the temperature reaches 55 degrees centigrade, it will impart brown color on the surface of the meat. So thereby what the perception of the people who are preparing it, so they will assume that the meat is perfectly cooked because the surface has been imparted with a brown color. Okay, so as far as the inside content or the inside of the meat is concerned, the temperature that has reached is extremely less. Okay, so that is the reason why it is appearing as pinkish in color. Okay, so this is in turn dangerous because the pathogenic bacteria are not exclusively killed. Okay, so this is about the premature browning. So the second one is the persistent pinkness. So as we have already discussed, the pH will in turn have an effect on the color characteristic of the meat. So the difference in the pH will also destabilizes the color of the meat. Okay, so that is about the cooking color problems existing in case of meat and how exactly they will impart the color change to your final product. Okay, so the next type of factor that we would be covering in today's lecture is vitamin E feeding to the cattle. Okay, so as long as an animal is fed with a vitamin E source, so the vitamin E content will remain in the muscle whenever an animal is subjected for slaughtering. Okay, so thereby it is preventing the oxidation and therefore the color of the meat is retained instead of undergoing any kind of a specific change in case of myoglobin state. Okay, so whenever vitamin E is fed to the livestock, so the increase in the alpha tocopherol concentration in the muscle is exclusively seen. As I've already told you, so it is an antioxidant which prevents oxidation. Okay, so the feeding of vitamin E content, specifically the animal which are exclusively reared for meat purpose, is of important concern. Okay, so whenever we are feeding a cattle or any kind of a livestock vitamin C, the vitamin E content will remain in the muscle. Okay, so as we have previously discussed, the chemical state of myoglobin is changing because of oxidation process. Okay, so since vitamin E is an antioxidant that will prevent oxidation process, thereby resulting in or maintaining or retaining the color of the meat. Okay, so the alpha tocopherol is an antioxidant that retards conversion of deoxymyoglobin and oxymyoglobin, which are the two different states of myoglobin, to metmyoglobin. So deoxymyoglobin will exist purplish in color, oxymyoglobin exists in cherry red color or red color, and metmyoglobin exists in case of brown color. Okay, so the feeding of animal exclusively with an antioxidant like vitamin E will decreases or prevents the conversion of these two myoglobin state to metmyoglobin. Okay, so this is the importance of feeding antioxidants to livestock as far as the color characteristic of meat is concerned. So the next in we have another important factor, that is wrapping of films. So whenever we are talking about wrapping of films, it is exclusively we are dealing with the type of packaging material that we are using for the packaging of meat. Okay, so let's have an understanding how packaging of a meat will have an influence on the color change or color characteristic of the meat. Okay, so herein I have established two important types of packaging films or the packaging material which are exclusively used for the packaging of meat as well as meat products, specifically the cut meats. Okay, so herein the vacuum packages. So the vacuum packages exclusively uses polyvinylidene chloride and polyvinyl chloride combination. Okay, so this is the combination which is exclusively used for vacuum packages of meat, okay, as well as the cut meat. So whenever such kind of combination of packaging materials are used, it is providing a barrier and protection against oxidation. So whenever it is providing a protection against oxygen, in turn it will result or it will further reduces or decreases the provision of occurrence of oxidation. So thereby the metmyoglobin formation will in turn be reduced. Okay, so this is the type of packaging material. So the next type of packaging material that I have given in here is the retail films. So the retail films exclusively uses polyvinyl chloride. Okay, so whenever such kind of retail films in case of meat is used, which is specifically shown in the picture 2, so it does not provide any kind of a permeability, it is a barrier to the oxygen. That means the packaging film will be permeable to the oxygen. So whenever such kind of packages are used, that means it is highly susceptible for oxidation, in turn resulting in the formation of metmyoglobin. Thereby the meat will appear dark brown in color. So this is the effect of wrapping films, okay, or the packaging material, and in turn the effect of color change that it is actually having. Alright, so moving on, the next important quality of meat that we will be studying is the water holding capacity. Okay, so whenever we are talking about the water holding capacity, it is advisable for everyone to understand the composition. So going back to your previous lectures wherein we have established the composition of the muscle, so the muscle will in turn contain approximately 75% of the water. So the water content in case of muscle is averaged to about 75%. Okay, so this particular most of the water is bound to the muscle fibers. So the muscle fibers here is myofibril. So this particular myofibril, that is the muscle fiber, will in turn imbibes water. That means it will hold the water between the muscles or between the cells. Okay, so the water will be entrapped between this particular cells. Okay, so that is about the water holding capacity. Okay, so before defining the water holding capacity, so we need to understand what are the different states of water that is existing in case of muscle. Okay, so herein I have given a specific figure which will establish what are the different classification of water in case of muscle. Okay, so the first one we have the bound water. The second in we have immobilized water or entrapped water, and next in we have free water content. So as far as bound water is concerned, so it is closely bound to the protein. Okay, in turn it will not facilitate any kind of a easy movement. Okay, and this particular quantity of water is contributing to about 0.5% alone. So herein you can see evidently from this particular picture, the water molecule are bound to the protein chain. Okay, so they are closely packed. So thereby it is not possible for freely move. Okay, so this type of water in case of muscle structure is contributing to about 0.5%. Okay, so the next thing we have immobilized water. So this immobilized water are the water which are held inside the structure of the muscle. Okay, so the only difference here is these immobilized water molecule are not held by any kind of a protein particles. Okay, so they are in the muscle structure, but they are not held by any kind of a protein structure. Okay, so this is the maximum amount of water that is exclusively seen in case of muscle, okay, which is contributing to about 80%. So next then we have free water. Okay, so this particular free water, as you can evidently see, so the water can easily move around the muscle. Okay, so this type of water is contributing to somewhere around 10% as far as the total muscle content water is concerned. Okay, so the water in case of muscle is existing in 3 different classes, that is bound water, immobilized water, as well as free water. As far as bound water is concerned, the water molecules are held by protein chain. Okay, so thereby it is not possible to freely move in the muscle. Okay, so second in we have immobilized water, which is contributing to the major portion of water in case of muscle. So these type of water will not be held by any kind of a protein structure or protein chain. So as far as free water is concerned, it contributes to somewhere around roughly 10%, and the water molecule can easily move around the muscle structure. Okay, so this is about the water holding capacity basics. Okay, so now let's just have an understanding on the definition of water holding capacity. So when we are talking about water holding capacity, it can be defined as the ability of the meat to retain its own water under any kind of an external pressure. So the external influences can be compression or any kind of a centrifugation. So the water has to be sufficiently held by the muscle. So that can be exclusively defined by the term water holding capacity. So that is defined as the amount of water that can be held by muscle. Okay, so that is about the water holding capacity. Okay, so the next thing, this property of the meat, that is the water holding capacity, is of important concern, because this is having an influence as far as the color, the texture, the juiciness of the meat is concerned. Okay, so if the water that is being held by the muscle is sufficiently enough, then it will impart a better chewable characteristic, better flavor characteristic, and better juiciness to your final product. Okay, so that is how important the water holding capacity of meat is concerned. Okay, so the fresh meat with good holding capacity is less prone to shrinkage. So if the water is sufficiently held together clearly, then it is susceptible, it is not or less prone to shrinkage or draining. Okay, so if the water is lost, the shrinkage of the meat can be effectively observed. Okay, so the water holding capacity of meat is very important as far as different processing operations are concerned, such as like cutting, grinding, filling, pressing, as well as heating. Okay, so these are the important factors which are affecting the water holding capacity. Okay, so further in I want to introduce to one of the important term that is regarded as drip loss. So the drip loss is one of the major challenge to the food industry. So most of the meat industry will try to avoid this particular drip loss in case of meat products. Okay, so what exactly this particular drip loss actually mean? Okay, so the drip loss, or it is also referred as purge, refers to the high protein fluid that is lost from fresh meat. Okay, so herein to explain this particular problem, that is drip loss, I have given a specific figure. So you have a cut meat and you have a specific portion of fluid next to it. So this portion of fluid is called as drip loss. Okay, so this portion of fluid is exclusively or extremely rich in protein and other kinds of minerals. Okay, so if the water holding capacity of muscle is sufficiently less, then it will result in the formation of drip loss. So that is the reason why the post-mortem changes of an animal plays a significant role. Okay, so if the animal processing after the slaughtering is not being done properly, then it will result in the formation of drip loss. Thereby, the industry will incur a lot of loss, because what happens whenever we are losing on the fluids, so effectively the shrinkage will occur, and thereby the weight of the meat will in turn be lost. Okay, so that is about the drip loss, which is one of the major and significant problem observed in case of meat industry. Okay, how exactly one can actually measure this particular drip loss? So this is effectively one of the change in the weight process. Okay, so the percentage of the meat before as well as after. Okay, so if the fluid which is lost, so of course the shrinkage will happen and the change in terms of weight will also be there. Okay, so difference in the weight will in turn gives us the measurement of the drip loss. So this is about the drip loss as far as water holding capacity is concerned. Okay, so this is one of the important and challenging aspect to most of the food industry to avoid the drip loss that is occurring in case of your meat product. Alright, so this is about the water holding capacity. Okay, so next thing we have another important factor which is affecting the color. So that is the juiciness. So how exactly juiciness is related? Okay, so the juiciness is said to be one of the important factor. Why juiciness is an important factor? Because it is having an influence on various sensory parameters as far as meat is concerned. So those sensory parameters includes the flavor, the texture, the chewability, and overall acceptability or overall sensory characteristic of meat as well as meat product. Okay, so all the things, that is the water holding capacity, is interrelated towards the juiciness. Okay, so higher the water holding capacity, the better will be the juiciness in case of your meat. Okay, so the juiciness of the meat is dependent on various factors. So what are these factors? So the first thing we have the amount of water retained in the cooked meat product. So if the amount of water that is retained in case of the cooked meat is higher, it will impart a better juiciness to your final product, and the consumer or the person who is eating it will get a better feel of this particular meat. So next then we have other important factor, that is the drip loss. So as I've already discussed, so the drip loss is the loss of fluid which extremely consists of rich in protein as well as other kinds of minerals. So if there is a drip loss and we are losing on a lot of fluid, so it will have an impact on the texture, the juiciness as well as the chewability of the final product. Okay, so that is the one of the important factor which is affecting the juiciness of the product. Okay, so next then we also have temperature of the cooking. So the temperature of cooking will also have an influence. So higher the temperature, greater will be the loss of fluid, and thereby the juiciness of the meat will be decreased, and it is vice versa. Okay, so these are the important factors which are actually affecting the juiciness as far as the meat quality is concerned. Alright, so this is about 3 important factors that we have studied in today's lecture. Okay, so to summarize what we have studied, so the eating quality of meat, so herein we have studied 3 important palatability characteristics, that is the color. What is the different types of color that is existing as far as myoglobin state is concerned? Okay, so how exactly the variation in types of color is occurring in case of meat? All of that we have covered in the particular color palatability characteristics. So the next thing we also studied about the water holding capacity. So how exactly the water holding capacity will in turn have an effect on the characteristic or the palatability characteristic of the meat, that also we have studied. Okay, so next thing we have also covered on juiciness. So what are the different factors that is contributing to the juiciness and how juiciness can be effectively maintained? So all of which we have covered in today's deliberation. Okay, so this is about today's class, guys. I hope everyone have understood about today's deliberation, and thank you so much.